Okay, so great. Thanks for having me, and it's great to give the first talk of RECOMB-Seq. And I will aim to do this live. So yes, it was a long time ago, but yeah, please bear with me. So I will talk about our work on spliced alignment of long transcriptomic reads together with Veli Mäkinen. And this work has recently been published in Bioinformatics. So let's see. So let's start with the biological context of this problem. So we have the genome containing a set of genes, and each gene contains a set of exons or coding blocks here in colored boxes. And each gene can consist of multiple copies. So here we have a gene consisting of 2 different copies, and there may be some mutations within the different copies. And a gene produce several transcripts, and they can produce several different transcripts depending on which of the coding blocks are included in the transcription. And we refer to this as the transcriptome, the full set of transcripts expressed by this genome. And in this talk, we'll look at long-read sequencing technologies, so PacBio, IsoSeq sequencing, or Oxford Nanopore. And the characteristics here is that these technologies have the capacity to sequence through the transcripts from end to end. So we get the full exon structure, and we may also be able to link mutations occurring at distant sites that shorter reads cannot do. However, these reads may, for some technologies, still contain more errors than Illumina read sequencing. So the bioinformatic problem here is that we want to map segments or exons of these reads down to the genome, to the correct sort of exon structure. So this is the bioinformatic problem. And why is this important? It's because alignments is sort of used as the fundamental step or the first step to many of downstream analysis, such as novel isoform detection and isoform quantification. So some of the challenges with this problem is, first of all, small exons or short exons. So here we see an example where we have a shorter exon that is well aligned in this example, but it may be difficult for other aligners. So they have sort of padded this exon onto either the downstream or upstream exon as an indel or an insertion in this case. And also there might be other problems such as very long introns, or if a gene exists in several different copies that are somewhat similar, this represents another problem. So there's a lot of previous work on splice alignment. So we have many short read aligners. We also have several long read aligners at this point, where Minimap2 and the SALT is 2 of the more popular ones. So many of these aligners, they focus on aligning the reads to the entire genome, which is of course great. You can find novel genes, but you do have to make some trade-off in the sort of the seed length or the index that you maintain in order to do this to become practically feasible. So in general, the good thing is that you can find novel and unannotated regions, but you do make some trade-off for shorter exons, because some exons might be shorter than your seed length or sort of roughly the seed length, which makes it difficult if there's an error occurring, etc. So our contribution is instead to focus on the annotated regions. So we have developed an algorithm and its implementation that we call ULTRA. That stands for something long transcriptomic read alignment with a U. What we do is that we focus the alignments to only annotated to and around annotated regions, and we show that we can do this more accurately. But we realized there's a big downside to not being able to annotate novel genes. So we can wrap our aligner around, for instance, Minimap2 to get the best of both discovering novel genes with using Minimap, while also having improved sensitivity in aligned in annotated regions. So So let's go through the method overview here. So as input, we take a set of long RNA-seq reads, which means usually PacBio ISO-seq sequencing or Oxford nanopore sequencing in FASTA or FASTQ format. And then we also take a genome in FASTA format and an annotation in GTF. And the output is common to other aligners. It's simply a SAM file with the genomic coordinates of the aligned reads. And so, okay, as for the algorithm, so I'll start going through the indexing or preparation step and then go through the alignment step. So in this figure, we see 3 different transcripts, and they in total contain 9 different exons. And what we do is that first we store something called parts, which is the union of overlapping exons. And we also store flanks, which we refer to as sort of a region downstream or upstream to the parts. And this could be either introns or the 3' or 5' UTRs. And finally, we store something called segments, which is that we cut these up into segments for each consecutive start and stop exon. And we also store exons. So these are the 4 different aspects or sequences that we store for the alignment. And the alignment itself is structured into 2 different parts. So for the first step, we use the parts and flanks here. And what we do is, so here's an example of 2 different reads. So what we do is we find maximal exact matches between the parts, flanks, and the reads. And of course, reads can have many different MEMS to several different regions. But let's say that these are the ones that hit this specific gene region that we have in the figure. And then we find the optimal solution to the collinear chaining problem, which is illustrated as an example here. And the solution, of course, a collinear chaining is defined by an increasing genomic coordinates and read coordinates simultaneously. So for instance, this is an example which is not collinear because these 2 MEMS occur in different orders in the reads and the references. Yeah, so we find this, and we have a theory paper about this in CPM. So, but this is again yet a fairly standard step in many read alignment problems. So now to the second step, we make use of the exon and annotations and the segments. So we can note that each of these MEMS in the optimal solution, they overlap segments and or exons in our indexing. So we take all the segments and exons that overlap on the genome, these MEMS, and we align them down back to the read. And then we solve a second chaining problem. And the characteristics here is slightly different to the common MEM chaining. So for instance, first of all, we need to allow for approximate matches because reads can contain errors and biological mutations. So we need to introduce some kind of weighting on how much we believe on the segment being aligned to this position in the read. And this is particularly true for like very short exons or segments that can map to many places. So this is something we introduce on weight. We also introduce overlap and gap penalties because it's not biologically meaningful in this solution to have overlapping segments or exons. And a final remark that I forgot to mention, we also include in this example very short exons that we didn't index. So there's some limit to what we can index as parts and flanks. So we solved this as a dynamic programming formulation, and the solution then that we trace back, we it defines the final genomic locations where we align the reads. So this is really the overview and what is being done. I want to mention a little bit about the time complexity here. So our MEM chaining is solved in nlog n. where n is the number of MEMS. But our segment chaining here, we did not get away with anything subquadratic. So it's quadratic in terms of the number of, if M here is the number of either segments or exons in the solution. And it wasn't trivial to solve this because of this, that we require weighted hits and also overlapping gap penalties. However, there is some interesting work in this direction where some authors have already shown that chaining with overlap and gap penalties can be solved subquadratically. And this, I think, was presented in HITSEC this year. So I encourage you to look into this paper if you're interested in this type of research. And so moving on to evaluation. So we tested our algorithm both as standalone tool and as a wrapper around Minimap2, where we use Minimap's alignments in unannotated regions, but our own in the annotated regions. We have compared it to Minimap2, both as by itself, and also it can take a guiding GTF annotation. And similar to, and we also evaluated it to the SALT with and without a GTF annotation. And in the paper, we looked at several different data sets. We looked at 2 different Oxford Nanopore sequencing data sets, 1 PacBio, and 3 different simulated data sets with various error rates. So in this presentation, I will only discuss 1 simulated data set and the PacBio Alzheimer's sequencing data set. And the results in these are will be sort of indicative to the rest of the data sets. So details are in the paper. So looking first at the simulated data set. So we took all the cDNA sequences from Ensemble, and we simulated read errors with about an 8 to 9 median error rate. And then we aligned them with these different algorithms. And we classify an alignment as correct if the aligner aligns to all of the exons that are present in the read with at most 15 nucleotides difference in the offset of splicing sites, meaning that we allow for some wiggle rooms due to read deletions, etc. We also looked at the more stringent modes, so details are in the paper. So we define this as correct if all the exons are found with a fewer offset than 15 base pairs. Inexact is then if at least one of the splice sites has a more 15 base pairs or more offset in at least one of the splicing sites. Exon difference is where either one exon is completely missing or in the alignment, or or a spurious exon is aligned to. Incorrect would be an incorrect genomic location, so the entire read is incorrectly aligned. Yes, so looking at the results here, we in general see that our aligner, both in standalone mode and as a wrapper, has higher accuracy than the second best performing aligner, which is the SALT using a GTF annotation. And we in general have less also than exon differences and incorrectly aligned reads. And yeah, so where is this increased sensitivity or accuracy come from? So in general, we see that, so what we did is that we took all the individual exons in each of the reads and we checked whether this specific exon was correct or incorrect. And then we divided it into exons of size 1 to 10 base pairs, 11 to 20, and so on. And then we looked simply at the total fraction of correct in this length class. So we can see that there is a big difference in the very short exons. And so this is really where we have the most improved accuracy, and then it sort of decreases or we become more even to other liners as the exon size increases. And we saw similar results for the other data sets with some specific things. So details are in the paper. As for the biological data set, then, so this is a PacBio Alzheimer data set, we do not have any ground truth alignment, so we can't really evaluate the accuracy. But we looked at the concordance between aligners, and we observed in general a high concordance in the positions of alignments. However, we do manually investigated several cases, and we observed that we are more sensitive specifically to small exons. So some specific examples I showed already in the early in the presentation was these short exons where we can see that here's our aligner, and we find this exon, and this is an already annotated exon that is well known, and there's a lot of reads aligning to this, while the other aligners has padded this as an insertion to the downstream exon. And this is sort of from an isoform that has been studied in relation to Alzheimer's. So we believe it's relevant for the Alzheimer dataset to sort of get these structures correct. There are many other such structures. Here's another one, a similar case. We also found novel combinations of exons, so which had the same thing. So this is this exon has been annotated, but the combination of exons are novel to other databases. So these are also some novel, interesting isoforms also related to, has been studied to Alzheimer's disease. And another class of errors is overfitting to canonical splice sites. So a canonical splice site usually is a GTAG pattern in the intron, start and end of introns. So we can see that our aligner stops the alignment here, while the other two aligners try to fit to this GT, immediate downstream intron. And they have to introduce an insertion here, which is an A. And then if we look at the downstream part, we instead align it here, while the other aligners fit then to the AG, which is this canonical downstream intron. So again, we don't know. The patient could have an insertion in the specific region, but just looking at the data, this seems like to be correct in this. And this has already also, this is an annotated, previously annotated splice site. So it's not like we're discovering anything novel here. So in general, the three different categories is that aligners usually have problems with aligning to small exons, overfitting due to alignment specific penalties for canonical splice sites. And these two I showed examples of. And the third one is the inability to align over some long or very long introns. So these are the general categories we see. As for runtime, our alignment algorithm is much slower when we have few reads. So if we look at only 200,000 reads, there's a lot of overhead generating MEMS, etc. We rely on a third-party library. So then we're much slower, 51 minutes compared to 10 minutes or even 6 minutes here. But as the number of reads decreases or increases, this overhead reduces, and it's eaten up by the total alignment time. So for instance, for biological data sets where we have 4 or 5 million long reads, we can see that we're about 2 times slower. So we take around 5 to 6 hours instead of 2.5 to 3 hours. And that's for peak memory usage. Minimap has the lowest memory footprint. We take about twice the memory. We are about similar for the largest data set. We have a similar memory footprint to the SALT, while Minimap is still the smallest footprint. So to sum up, I've described a splice aligner for transcriptomic long reads. It shows higher accuracy, particularly for short exons, but also because it's not using any penalties, alignment penalties to optimize for junctions, to align to canonical junctions. And also we can use our aligner as a wrapper around Minimap2, so we can both do the novel gene. It can be used as an end-to-end aligner to both detect novel genes and to get more accurate or sensitive results around the annotated gene regions. And for future work, we aim to improve both speed and peak memory, of course, And also related to this is to remove this third-party mem finder to remove intermediate files, etc. So the tool is available on GitHub, installable through Conda, and paper just got out in bioinformatics. And I will stop there and happy to take any questions.
Thanks, Kristoffer. So if any of the listeners have questions, then please use the question and answer tab to type in your question. I think we have like time maybe for 1 or 2 quick questions. But maybe while we wait, if somebody has a question, then I could ask that. So you said that you can use Ultra as a wrapper with Minimap. So is it possible to use it with some other aligner, or is there some fundamental reason why not?
No, there's no fundamental reason. There's just a small implementation specific reason now. I mean, we only currently have a function for calling Minimap, etc. But you can plug in any other aligner, such as the Salt, for instance. This just requires a small additional function to be implemented, where we call this aligner instead.
Okay, great. I still see no open questions, and since the time is pretty much up, then maybe we move to the next talk.